PChem problem solvers or PChem problem survivors, depending on the minute. We're doing the Coalert test research group, and a little bit of review from the previous Chalk Talk. The current test for staff also fluoresces in the presence of E. coli, and the purpose of this research is to find a fluorescence that only indicates staph. The previous lab group geniusly found that glutamic acid is unique to staph, so the primary goal is to find a dye that fluoresces in the presence of glutamic acid. The last lab group also suggested that we find something that has a primary amine so that it can form a covalent bond with glutamic acid. There will be spoilers about that later, but the two primary dyes that we'll be looking at in this research are the 2,7-dichlorofluorescein and the rhodamine 110, but we will also be discussing what the previous lab group found about Congo red and rhodamine B. So basically, in this particular experiment, we're emphasizing the protocol necessary for each of these dyes and how to proceed forward.
So the previous group told us to try rhodamine B, or actually they tried rhodamine B themselves and didn't end up working, so then we decided to try Congo red because it had an amine group that was compatible with our glutamic acid, and we found out that Congo red is actually a pH indicator and did not work for our experiment. So we moved on and tried to find other dyes that would work for us, and we came upon 2,7-dichlorofluorescein and rhodamine 110. We ended up trying both of these in one lab period and made a stock solution and diluted them. They're actually really both super fluorescent, so they were a bright green color. And then went to run the wavelength versus absorbance and wavelength versus fluorescent graphs.
So basically our procedure for this, we followed pretty closely what the other lab group had done. We basically did 3 mils of a dye solution, which we did by diluting 100 and then 500. And then we did 1 mil of water and then slowly decreased the water and increased the glutamic acid to avoid actually diluting our solution but still increasing glutamic acid. But what we found actually in creating our stock solutions was that that procedure isn't necessarily like streamlinable, because rhodamine 110 wouldn't fully dissolve even in like 1,000 milliliters, and that 2,7-dichlorofluorescein was going to be way more dilute at 500. So we ended up just trying to find a good absorbency that would register in the UV vis at about half. And then we just kind of compared them and made sure that they would come up at about the same fluorescence as well. And so that's kind of our general procedure, as well as like how we overcame some of the problems that we encountered. And we're actually going to be kind of working backwards from there, coming up with concentrations from our data that we collected via UV vis and fluorometer. So.
When we measured our solutions in the fluorometer, we came up with 4 graphs: an absorbance versus wavelength and a fluorescence versus wavelength for both 2,7-dichlorofluorescein and rhodamine 110. We compared the fluorescence versus wavelength for both compounds, and we thought that this was the most significant graph in helping us establish the purpose of our experiment. So this curve right here is the stock solution. So we ran the stock solution of our compound, specifically for this graph, 2,7-dichlorofluorescein. And then the rest of the curves are increasing concentrations of glutamic acid and decreasing concentrations of water. And as the graph shows, the curves decreased significantly as glutamic acid increased, which indicates that 2,7-dichlorofluorescein was quenched the most by glutamic acid. Therefore, 2,7-dichlorofluorescein is a better compound dye to use than rhodamine 110.
And so moving forward with this experiment, we recommend yet again that it would be another procedure refinement type, I guess, furthering this experiment. We recommend that 2,7-dichlorofluorescein is used just because it was quenched the most in comparison to rhodamine 110. We also found that when using the graphs, or when interpreting the graphs, it was quenched more, and that was reflected in our data as well. We also found that it wasn't necessary for there to be a primary amine group for our indicator dyes as well. We also just want to make sure you don't pick a pH indicating dye, which is what we did with Congo Red as well. And another thing that we also had some troubles with was that when we diluted our rhodamine 110 and 2,7-dichlorofluorescein as well as Congo Red and the other solutions, we kept the serial dilutions constant. So we diluted them into 100 milliliters and then diluted 1 milliliter of that solution into 500 milliliters. They weren't completely soluble in water, so what we were thinking is that you might have to work backwards from your stock solution to find the correct concentration, just because different compounds they have different solubility in water. So that will have to be something that you look into, just because that will give you better fluorescence and better absorbance readings when you run the solutions in the fluorometer. We also had really good success with the concentrations and the contents of our cuvettes. We kept the concentrate, or the volume of our stock solutions constant in all 6 of our cuvettes, one being an exception being the blank with just DI water. We kept 3 milliliters of our stock solution, and then we changed the concentration of glutamic acid as well as water in our cuvettes. We found that gave the most consistent results with one another. So those are our kind of hints for you guys, just to kind of help you further along your procedure. And best of luck, and as always, smash that like button.